Bismillahirrahmanirrahim. Today in this lecture I am going to discuss the important disease Psittacosis or ornithosis. In this lecture I will cover the definition, the causative agent, the transmission, life cycle, sign and symptom, epidemiology, diagnosis, treatment and prevention of psittacosis. First of all, I am going to define that what is psittacosis. Actually, psittacosis is a form of pneumonia that usually causes fever. You should note that psittacosis is a form of atypical pneumonia that usually causes fever, coughing, headache, and chills. Subclinical infections are very common and stress appear to enhance the susceptibility to the disease. Disorientation or even delirium in some cases indicate that the nervous system can be involved. Now the question arises that why this disease, this form of pneumonia is called psittacosis. Actually, the term psittacosis is derived from the disease association with the psittacine birds. What are psittacine birds? Psittacine birds are actually they are uh, parakeets and other ferrets. Due to the association of disease with parakeets and other parrots, the disease's term is psittacosis. But later it was found that the disease can also be contracted from many other birds such as pigeons, chickens, ducks, and turkeys. Therefore, the more general terms ornithosis is also into use. Now, if we talk about the causative agent of this disease, the causative agent is Chlamydia psittaci. Chlamydia psittaci is gram-negative, obligate intracellular bacterium. The taxonomy of this organism has recently been revised. The genus name has been changed from Chlamydia to Chlamydophila. This taxonomic changes has also been made with Chlamydia pneumonia. One way now you know that rickets is also intracellular parasite while chlamydia is also intracellular parasite. One way chlamydia different from rickettsias which are also obligate intracellular bacteria. Chlamydia different from rickettsia because it form tiny elementary bodies is one part of their life cycle. Unlike rickettsias, elementary body are resistant to environmental stress, therefore they can be transmitted through air and do not require a bite to transfer the infective agents directly from one host to another. In the previous lecture, we have discussed that rickettsia are transmitted by the bite of the various vectors are required, but in the case of chlamydia there is no need of vector. Now if we talk about the elementary body, look at this figure. This is the elementary body. The elementary body is then convert to reticulate body and intermediate body. We are discussing the what is elementary body, reticulate body and intermediate body in the next slide. Now, if we talk about the transmission of this disease, the disease is seldom transmitted from one human to another, but usually the the human to human infection are rare, but is usually spread by the contact with the dropping and other exude of the fowl. One of the most common mode of transmission is the inhalation of dried particle from the dropping. The birds themselves usually have diarrhea, ruffled feathers, respiratory illness, and a generally dropsy appearance. These are parakeets, and parakeets and other parrots sold commercially are usually but not always free of the disease. Many birds carry these birds. Many birds carry the pathogen in their spleen without symptoms, becoming ill only when stressed. pet store employees and people involved in raising turkeys are at greatest risk of contracting the disease. Now, what is the life cycle? Look at this figure. Uh, Elementary body attached to the host cells, and this is the host cell, and this is the nucleus of the host cell. Elementary body attached to the host cells. The host cell phagocytizes the elementary body in the vacuoles. This is vacuole forming of vacuoles. The elementary body reorganizes to form reticulate body. This is the reticulate body. Now the reticulate body divide and produce multiple reticulate bodies. Reticulate body convert back to the elementary bodies, and now the elementary body release from the host and attack on the other host. Now, if we talk about the sign and symptom, chlamydia infect primarily epithelial cell of the mucous membrane or the lungs. They rarely cause invasive disseminated infections. Chlamydia psittacosis infect the lung primarily. The infection may be asymptomatic, detected only by rising antibody titer, or may produce high fever and pneumonia. Human psittacosis is not generally communicable. Now, if we talk about the diagnosis, first of all, I am going to discuss that chlamydia cytosis infect birds. Uh, Human are infected primarily by the inhaling of organism and dried bird feces. Laboratory diagnosis. Chlamydia form the cytoplasmic inclusion, which can be seen with special stain, gems or stain. Look at this figure, or by the immunofluorescence. Look at this figure. These are the cytoplasmic inclusion. The gram stain is not useful in exudate. The organism can be identified within epithelial cells by fluorescent antibody staining or hybridization with rDNA proofs. Chlamydia antigen can also be detected in the exudates. Exudates are urine by enzyme linked immune servant assay ELISA. Chlamydia can also be grown in cell culture treated with cyclohexamide, which inhibit host cell but not chlamydial protein synthesis, thereby enhancing chlamydial replication. Serological tests are used to diagnose infection by Chlamydia psittacosis and Chlamydia pneumonia, but are rarely helpful in diagnosing the disease caused by the trachomatis. Only you can diagnose the serological test by serological test the Chlamydia psittacosis and pneumonia, but rarely helpful in the diagnosis of the Chlamydia trachomatis. Why? Because the frequency of infection is so high that many people already have antibodies. All chlamydia share a group-specific lipopolysaccharide antigen, which is detected by complement fixation test. What is complement fixation test? We will discuss it later. Actually, they also possess species-specific and immunotype-specific antigen or protein, which are detected by immunofluorescence. Chlamydia psittacosis and Chlamydia pneumonia each have one immunotype, while Chlamydia trachomatis has at least 15 immunotypes. Now, what is the treatment and prevention? Now, all chlamydia are susceptible to tetracycline, such as doxycycline, and macrolides, such as erythromycin and azithromycin. The drug of choice, you should note that the drug of choice for chlamydia psittacosis and chlamydia pneumonia infection and for lymphogranuloma verorum is tetracycline, such as doxycycline. If we talk about the prevention, there is no vaccine against any chlamydial disease. Psittacosis in human is controlled by restriction, restricting the importation of cytokine birds. These are cytokine birds, destroying 6 birds and adding tetracycline to their feed. Domestic flock of turkeys and ducks are tested for the presence of psittacosis. In the next lecture, I will discuss about the pneumonia, chlamydia pneumonia.